Alright, so this is my bi-weekly update, a belated one that was supposed to have been done on Friday of last week, but here it is now. Okay, so regarding progress, I started, as mentioned in my last meeting, docking GD2 onto the Chimera 1 homology model present within OneDrive. And this included not only the Chimera. I started with the Chimera, but I docked it onto the 5 structures that were in there, the 5 PDBs. So I completed the dockings using the standard pre-Teleris 2013 protocol that Ray and I developed for the phytochemical screening. Before doing the dockings, I read in the Design of Ligand Binding Sites paper that it was advisable to relax the structures. And relaxing essentially minimizes the structures in a way that makes them compatible with Rosetta's force field. And they advised us to do this in the case that the structures didn't originate in Rosetta. And they didn't. They came from AlphaFold, so I thought this was prudent to relax them and ensure that they wouldn't have any clashes due to Rosetta's energy force field. So I docked GD2 into all 5 structures, all 5 PDB structures. My docking protocol involved selecting 3 coordinates in the CDR regions of each of the structures and generating 1,000 models. So 1,000 docked poses were generated, and the top 10 of those were selected in the final screening, and those top 10 are the ones that I have in HPCC right now. Top 10 for each protein structure. And I haven't exactly looked at the scores yet, but I think I will select the best 1 or maybe 2 of those based on binding energy score, and then those will be used to continue the workflow. And namely, the workflow in continuation onto this 4th point. We'll use Glycan Dock for refining the best docked poses obtained from the initial dockings, the top 1, top 2, one of those. And then after Glycan Dock refines those, the single best pose obtained by Glycan Dock for each of the protein structures will then be designed or affinity matured in terms of mutations. So affinity maturation will be conducted using the Rosetta and Design of Ligand Binding Sites protocol. Initially, a while ago, I was considering SnugDock to, further optimize the homology model in Rosetta, because SnugDock was intended for docking ligands, or not ligands, protein ligands specifically, into homology models. It was tailored to reduce the inaccuracies of homology models, but I'm no longer considering it because it only works on protein ligands. It doesn't work on standard ligands, carbohydrates, small molecules. It only works on proteins. So only for protein dockings is it actually successful. And I tried it. I tried it using the Rosie web server, and it gave me that error. So a bit about the homology models, which I touched on during our meeting, but I believe they will be made clearer with these figures that I prepared on the slide. So the homology models produced in AlphaFold, the 5 that are currently in the folder on OneDrive, they're a bit strange, particularly because they have this abnormally long linker connecting one of the termini to the other in the CDR region, and they also have an abnormally long terminus here. And I noticed in some of the poses, not all of them, but a few, that the terminus, shown in figure 1 to be this protrusion on the right, sort of assists the ligand in its docked pose. And this is not the best example, but in some of them the ligand is almost fully reclined against the terminus, which was concerning because that terminus likely isn't shaped like that in the single fab. That's probably part of a complex, or probably due to some inaccuracy in the AlphaFold modeling. So as you said, perhaps I should redo these dockings by trimming off the abnormally long terminus. And I found some justification for this. So chopping it off actually seems pretty reasonable to me, because apparently AlphaFold outputs confidence values for all of the predicted regions. I haven't seen them yet, so I'd have to see it to actually verify that this region has a low confidence value, the protrusion in the massive linker. But if it's this PLDDT value is below 50, this website, which I cited here on AlphaFold, says that they often have a ribbon-like appearance, which they do, as shown in Figure 2, the long linker and the terminus, and they shouldn't be interpreted. So they're low confidence, they're likely inaccurate. So I presume, yeah, chopping them off, as you mentioned, is a good idea. I have not done this yet, but in the final run of things, I definitely will. Currently, I'm just beginning the dockings of GD2 and the whole workflow, really to tweak it for now and see how it pans out in the future. So I also, well, I mentioned this previously, I did relax the protein structures. After relaxing all of the 5, I docked them using pre-Talaris 2013. I mentioned that currently 10 poses are being selected. However, I'm thinking of transferring just 1 or 2, maybe, to the Glycan Dock stage, which is the subsequent stage. So out of the 1,000 poses that are generated with pre-Talaris 2013, instead of the top 10 being selected for each protein ligand pair, I will select the top 1 or 2. Possibly. This is arbitrary. I don't really have justification for it, but for now that simplifies the workflow. Maybe more will be included. If more will be included, that likely will be better. So then Glycan Dock will be run to refine the pose. And what Glycan Dock is best visualized in this thumbnail here. So it takes an input of a protein ligand complex and it refines it such that it better matches a native structure. There's greater correspondence. It just optimizes its position to more properly resemble an actual docked pose. So it needs a protein ligand complex as an input, in that complex we will obtain with the standard pre-Talaris 2013 docking. And then the best docked poses obtained with Glycan Dock. Again, the single best docked pose obtained with Glycan Dock for each protein ligand pair, those will be fed into the affinity maturation or the design workflow. And then the design of ligand binding sites paper advises users to not just do 1 round of design, to do several if needed. And they say this until you can see a convergence towards a lower and lower energy score. Yeah, you should conduct multiple rounds of design. I don't know how many rounds it's going to take, but I'm going to start with 3. Hopefully it won't be too many. Hopefully there's some energy minimum that the ligand position is converging towards. That will be ideal. But that can only really be tested if I do more than 1 round. So I'll start with 3. Maybe it will increase by the end. So this is a very simple and crude flowchart of the computational workflow. So the sequence building step, that is accomplished using the spreadsheet, the 10x genomics spreadsheet, in which the variable regions, in the case of chimeras, both the frameworks and the CDRs, and in the case of the grafts, only the CDRs, are put onto and are used to generate full-length fabs. So those sequences will be built using a MATLAB script that I am developing. Those sequences, once fully assembled, will be subject to stability optimization and calculations using DDG monomer. And Nathaniel's focusing on that heavily now. Once the stability calculations are conducted and the AlphaFold models are generated, the models will be introduced into Rosetta. Initially, they'll be relaxed to make them fit with the force field. If needed, their large terminus will be trimmed, since hopefully that doesn't impact the docking. Then, or maybe there's a means to get rid of it in AlphaFold. That will be preferable. Then the standard pre-Talaris 2013 docking will be conducted. and the best 1 or 2 poses will be selected for each protein ligand pair. Glycan Dock will be used to refine that best 1 or 2 poses, and the best Glycan Dock pose will be selected for each protein ligand pair. Those will go to affinity maturation, and multiple rounds of design will be conducted, 3. And then the sequences from the affinity maturation that yield a binding energy score. I'll have to decide on a threshold. Obviously, the threshold is going to have to be below the binding energy score of both Glycan Dock and the standard protein ligand docking. So I'm going to have to pick a interface score that's, I guess, significantly below. Maybe, I don't know, maybe I can do like 1 or 2 standard deviations below the average of Glycan Dock score. And those sequences will be selected for library expression. So currently the most important aims is automating this whole process. Namely, my main considerations are ensuring that CDR regions are automatically detected, and then the coordinates of that general area is isolated and the initial docking is conducted there. From what I've seen online, AutoDock VNA might make this easier, particularly because it makes global docking easier, as shown with this grid-based approach in this GUI thumbnail. The scripts are far easier to run than in Rosetta. You do still specify an initial coordinate and a grid size, but it's far easier to run, and I'm thinking that it's also far easier to automate because there's far fewer components involved. But for now, this is just a consideration. It's not really a priority. So for now, I'm focusing on doing this in Rosetta. And to do this, I suppose that I'll use the detect interface.py. Once the protein ligand complex is formed after the Glycan Dock, detect interface.py will be used to select the residues that will be mutated in the affinity maturation. Those will all go into a res file, which is needed as an input, and then those will be mutated with all 20 canonical amino acids to find mutations that improve the binding affinity. So this script, I'm hopeful, will play well into the automation stage. Regarding automating the CDR finding for the initial positioning of the ligand in the standard pre-Talaris 2013 docking, I'm still considering multiple options. Maybe there's like a motif recognition script somewhere, and I could use a framework region, or no, something X angstroms away from a constant region close to the CDRs that will be considered the initial starting coordinate. But for now, I mean, I'm doing it manually, and it works fine with just finding the CDRs manually, selecting a residue on them, and using that as the initial starting coordinate. For now, that works fine. Okay, so I looked at, as requested, the numbering system used in the 10x genomics system spreadsheet. And what I found is that Chothia and Kabat don't really add up too well. I haven't really looked at IMGT because I haven't really found a web server that recognizes CDRs and aligns them to a specific numbering system. So this is the sequence I used. It's clonotype 1, the variable heavy region. This can be copied and analyzed whenever. So I tried it on this, on a specific web server linked here at the bottom of this slide. And I submitted the sequence, and it spit out these CDRs when I used the Kabat numbering system. And they did not line up to the actual CDRs listed in the spreadsheet at all. So for this one and for a few others that I tried, Kabat didn't really work. I don't know if it's either a problem with the web server, the length of the sequence, or something else. I'm not ready to disconsider Kabat, but these are the initial results I found. Chothia yielded much better results. CDR1 and CDR2 were exactly what they actually were in the spreadsheet, and CDR3 was a little off. It was missing a CAR at the beginning, but it was much better than Kabat. IMGT, I don't know. I need to find a web server that can do IMGT. I haven't really found one yet. But obviously this is imperative because without knowing the proper numbering system, the grafts we will perform using the spreadsheet and the sequence building we'll perform will kind of be in vain. So onto the sequence building. I'm using a MATLAB script that I'm developing and some Excel to assemble the sequences from the 10x genomics data, to putting the variable regions of the chimeric and the grafts into the rest of the fab. And I hope to have this ready by Tuesday when we have our sequence-based meeting. So the script that takes all of that and assembles it into the full fab. And yeah, I've had better results with Chothia than Kabat, but I want to be really sure that I know what numbering system is used in the 10x genomics spreadsheet, so the grafts are actually done properly. And yeah, this was as mentioned previously, the link to the web server that I used. And in order to help me learn more about numbering systems, I'm reading this paper. It goes through Kabat, IMGT, Chothia, Martin, and I think a few other numbering systems. And it goes into details about their differences and how they consider certain motifs. And yeah, that's about it.